Okay, well, if you're still watching, I guess it means you're either making you watch in class or it's helping you. So I hope this is helping. Um, so yeah, we were looking at this picture of how introns are removed, exons are spliced together, and so yes, this mature mRNA is ready to be transported out of the nucleus into the cytoplasm for translation. Um, Okay. Oh, sometimes you'll see these models. Same thing here, it's just more the same. DNA makes pre-mRNA, RNA is processed. Mature mRNA floats out into the cytoplasm for translation. Um, this processing, there is GTP, G with 3 phosphates added to an end. Sometimes you get these what are called UTRs or untranslated regions stuck in there. Hopefully you're familiar with what the start and stop codons are. Here's some more of that untranslated regions, and then a poly A, a lot of adenine on the tail. Yeah, these caps, and it's just there to protect it. Really, this is the piece that's going to be used for transcription. This helps it make its journey, if you will, through the cytoplasm. Just helps it kind of maintain its integrity as it starts to touch and come into contact with everything else floating around in there. Okay, now we will spend a lot of time on this, and we'll do a few labs where we can turn on the promoter region of DNA. So really I just kind of wanted you to have this idea that out in front of the part, where transcription happens, kind of how the enzymes know to float in and make the right size bubble, or to make the bubble in the right spot, to make the fork in the right spot for transcription, depends on these promoter regions. So just think of the promoter region as just something out in front of where transcription is going to happen. But promoter region, promoter region. The promoter is a part of the DNA. The promoter is a region of DNA. That really helps as we move along. Okay, once again, another one of my favorite models. The thing I really like about this is you do kind of get that model of how, okay, here's how the pre-mRNA gets shortened and capped off. But the thing I really like about this is that a cell can take the same primary transcript, in other words, the same DNA was used, but make a different hormone, a different protein. So here a thyroid tissue making a hormone that regulates calcium. Here's brain tissue, brain cells, taking the same DNA, the same mRNA, processing it differently, and as a result a different protein is translated. And this protein is functions as a pain receptor. So, I don't know, to me that's fascinating. Same DNA, same mRNA, pre-mRNA, different processing, as a result, different protein being made. Okay, so we've done kind of replication, you kind of understand some of the important aspects of transcription. Now let's look at the final piece, translation. And really, you've done a lot of translation, or at least played the letter game when you were using this chart. And I think we'll pick up here, because you guys really already do know what the wobble position is. Yeah, we got time. The wobble position is that third spot. So technically, it's the third spot on the tRNA that the mRNA sticks to. But you guys knew if I said, what can make valine? Well, GUU, GUC, GUA. I mean, GU is all you needed, and then that third position really didn't matter. And that's the case for most of these. That third position can be 2 different things, 4 different things. But yeah, the fact that the third position can change, that's what we call the wobble position. And it was actually discovered by Dr. Wobble, not the fact that it can wobble and change, but just kind of ironic that his name does also somewhat describe the properties of that piece.